Good morning, everyone. Today we're going to look at mitosis and meiosis. So far we've talked a little bit about the characteristics of life. We've talked about molecules that are required to sustain life. And most recently, we've talked about the structure of the cell, okay, the cell membrane, the internal compartments. Today we're going to look at how new cells are made, okay? As you know, new cells need to be generated throughout the course of an organism's life. Like most of our cells do not have an infinite lifespan, and therefore new cells must be created. Okay, so new cells are created for different purposes, for growth and development, so basic maintenance throughout an organism's life, and then also for reproduction. So mitosis and meiosis are two types of cell division that takes place in order to regenerate and produce new cells. And so we'll start this discussion by understanding that pre-existing cells divide to form new cells, okay? And these new cells that are being made are important in order for an organism to continue to grow, to repair damage, and most importantly, for reproduction, okay? So the cells contain genetic information in the form of DNA. We're clear about that. When a cell divides, the information in the DNA must be accurately replicated so that it can be transmitted to the daughter cells, okay? So we're going to walk through the processes that are necessary for new cells to be formed and what actually needs to happen during these preparation stages in order for the daughter cells, okay? The daughter cells are the new cells that are being formed. In order for these daughter cells to get the accurate genetic information passed down through them, okay? And so we know that the DNA houses, the nucleus houses the DNA, all right? This DNA is organized into packages that we'll call chromosomes, all right? You've heard about chromosomes before, all right? These chromosomes are going to be, again, the structural units that are passed down from one generation to the next. Mitosis is the form of cell division where the parent cell transmits information down to the daughter cell in an exact copy, okay? So daughter cells that are created through mitosis are going to be genetically identical to the parent cell, whereas meiosis is a series of steps that leads to a reduction in the number of chromosomes, and each daughter cell that is created is going to be genetically different, right? So these are going to be two totally different processes with totally different purposes. So in your notes, you should jot down really quickly that the purpose of mitosis is to produce new body cells, somatic cells, okay, that will facilitate growth and development throughout an organism's life to repair damage and so forth. While meiosis has a totally different purpose, which is reproduction, okay? This is where our gametes are formed, the sex cells, all right? So the chromosomes, we know, are the major carriers of genetic information. These are going to be found in the nucleus, and they're typically going to be associated with proteins wrapped around them. The human cell has 23 pairs of chromosomes. So in a normal individual, our somatic cells are going to have 23 pairs of chromosomes, which is going to total 46, okay? These 23 pair of chromosomes, you're going to get a set from mom, you're going to get a set of chromosomes from dad, okay? So you're going to get 23 from mom, you get 23 from dad, and that together is going to be our 46 chromosomes that are found in our somatic cells. This image right here is a karyotype, right? This karyotype is simply a layout of all of the chromosomes that are found in the organism, okay? For the human, this is a human karyotype, and it shows you the identity of each of the chromosomes that are found in humans, and they are identified based on numbers. So we have numbers, chromosome numbers 1 through 22. And as you can see, they exist in pairs. And then we have the pair of sex chromosomes that are either going to be XX or XY, which will determine the sex of the human. And so these chromosomes 1 through 22 in our sex chromosomes, there's been an effort to investigate which genes are encoded on which chromosomes and which characteristics. And so there's the Human Genome Project. Write down the Human Genome Project, and when you get a moment, go out and read about it. So this was a very very interesting undertaking for scientists to go out and map the human genome. And I think at this point we have mapped all of the genes that are found in humans. We know which genes are on which chromosomes, which characteristics they're associated with, which traits, and so forth. And so it's a pretty awesome project. So do go out and read about the Human Genome Project, okay? And so the point we're making here is that cell division is a very important process in terms of maintaining an organism throughout its life and reproduction. And then we're clear that this process needs to happen in a very orderly and in a very trustworthy manner. We don't want to compromise the fidelity of this process. And so, okay, so going back to my point about the Human Genome Project, this effort was to, again, identify all of the genes that are found in humans. And we know that genes are just sequences or segments of DNA that code for a functional protein product. And these genes are considered to be, informational units. That's what we'll refer to them as, because they provide the information for cells to carry out their functions in the form of coding for proteins. Okay, and so genes are responsible for traits and characteristics that we see in an individual, such as eye color, the winglet of a fly, the seed color of plants. So these genes are very important instructional units, all right? And these genes are what's being passed down from one generation to the next. And so the one point that I'll make here that you should note is that these chromosomes, so the human has 46 chromosomes in our somatic cells, but the number of chromosomes can differ amongst various species, okay? Information contained on the chromosomes can also differ, okay? So while the human has 46 chromosomes, the number of chromosomes that might be found in mice or say in a domestic cat can differ, okay? So do understand that. Okay, so let's look at two things today. We want to understand the stages of the eukaryotic cell cycle, and we want to be able to describe what events take place in the cell cycle. Okay, so we've already kind of made the point that when cells reach a certain size, they're either going to stop growing or they're going to divide to make new cells. Okay, not all cells will divide. All right, we'll take a look at the stages in the eukaryotic cell cycle, which basically prepares the cell for division, and then the actual cytoplasm divides through cytokinesis, and we have a new daughter cell being made. All right, so this cell cycle can take anywhere from 8 to 20 hours in plants and animals. It just depends. So let's take a look at the cell cycle. Okay, in your notes, you should indicate that there are two main phases of the cell cycle. Okay, two main phases. There's interphase, and then there's M phase. Okay, interphase, if you look at our schematic right here, kind of looks like a clock. Okay, the cell spends most of its time in interphase. Okay, so if you think about a cell that's getting ready to prepare for division, what needs to happen? There's a lot of preparation that goes in to ensure that all the things have happened the way that they should before the actual cell division takes place, which takes place during this M phase. Okay, this is where mitosis takes place. Okay, so the interphase, okay, is the preparation phase. This is where the cell is growing and obtaining nutrients, duplicating its DNA. There's a lot happening during interphase. So suffice it to say that the cell spends majority of its time in interphase preparing for division, and then division actually takes place during M phase, okay? So let's take a deeper look at interphase, okay? Most of the cell's life is spent in interphase, okay? The one point to note here is that there's no cellular division taking place during interphase. This is simply a preparation stage, okay? So during interphase, the cell is growing, okay, in our G1 phase. In S phase, it's synthesizing the materials that it needs to prepare for division, okay? Synthesizing new DNA, all right? Because we want these daughter cells to have the same exact genetic content that this parent cell has. And so we need to duplicate the DNA that's already present, okay? And then G2, it's again another gap phase to prepare and do checkpoints to make sure that everything has happened with fidelity, okay, before it progresses on to M phase, okay? So we're saying that interphase is a very important preparatory phase in the eukaryotic cell cycle, where the cell is growing, synthesizing the things that it's needing, preparing for division. But what is not happening during interphase is cellular division. So G1, let's take a look. We're diving deeper into the steps of interphase. Interphase is characterized by 3 steps or 3 phases. G1, gap phase 1, okay? This is where this is an interval of growth, okay? There's no DNA synthesis occurring. This is just normal growth and metabolism taking place here at G1. The cell is getting all the things that it needs in order to prepare for division. G1 is typically the longest phase of the cell cycle, and the enzymes that are required for DNA synthesis are becoming more active, okay? It's preparing the cell to enter S phase, okay? So G1 is a period of growth and normal metabolism, just normal cellular activity, and enzymes that are required for replication are becoming active, okay? So we're going through the phases of interphase. So we have G1 and we have S. S phase is a synthesis phase, okay? Synthesis means to make something, right? So during this phase, we're making new DNA. The DNA that's present in the cell is going to have to be replicated, okay? So again, you imagine with me that we have one cell and we're trying to duplicate this cell. We're trying to make an exact copy of the cell that we started with, okay? So if we started with the cell that has 46 chromosomes, our daughter cell, the new cell that we make, needs to also have 46 chromosomes, okay? So in order for that to happen, we've got to replicate this DNA, okay? And that's going to happen during S phase, okay? Once this duplication is complete, cells will enter another gap phase, and that's gap phase 2, G2, okay? G2 phase is where this cell is preparing for division now, okay? This is a relatively short phase. Again, it's gone through its checkpoints, make sure everything has happened the way that it's supposed to happen, and then it can progress on to M phase. So let's look back at our schematic here. Interphase, we have G1 phase, okay, that period of normal growth and metabolism for the cell. Okay, we have S phase, right, where DNA is being replicated, right? Because again, the goal here is to create a new daughter cell that's going to have the exact genetic content of DNA. And then we're going to go on to gap phase 2, which again is our last sort of checkpoint to make sure that things have happened the way that they need to before we go into M phase. So let's look and see what happens at M phase. So M phase involves 2 main processes, okay? Mitosis and cytokinesis, all right? Mitosis, okay, I know it's easy we use that term mitosis and we think that that's the term that refers to cell division, but it's not. The actual term that refers to the cell division, the splitting of that cytoplasm into 2 new cells, that's going to be called cytokinesis. Cytokinesis is the actual division of the cell cytoplasm, which is going to basically be a splitting off, if you will, into 2 new cells. So we start with 1 and we end up with 2 genetically identical cells being created as a result of mitosis. Okay, and so mitosis is the nuclear division. Okay, it's producing the two nuclei, a set of steps that leads to these two independent organizations of our chromosomes preparing for separation into two new cells. So again, we have a schematic right here that represents what's happening. Now this one is going to go in the opposite orientation. And so do get familiar as you're reading through your text and maybe some online resources that the representation of what happens during the cell cycle can look a little different, but the same thing is happening. So in this schematic here, we're going to go counterclockwise. So we have our G1 phase, right? And what's happening at G1? All right, G1 is our period of growth and metabolism for the cell. Okay, the cell wants to divide into a new cell. So G1, we're going to have our period of growth and metabolism. S phase is our synthesis phase. What's happening at S phase? Okay, new DNA is being made. All right, so we're going to duplicate or replicate our DNA during S phase. Okay, G2 is our sort of final preparation stage for the cellular division. Okay, gap phase 2, and then we have M phase. Okay, M phase is a very orderly step-by-step process that's going to lead to cytokinesis, where that cell actually splits off into two new cells. So we're going to take a moment now to look at the steps of mitosis. So M phase is characterized by mitosis, okay, and then cytokinesis, where this cell actually divides off into two new cells. All right, so let's take a look at the steps of mitosis. Okay, mitosis is a part of M phase, and it takes place over 5 stages. Okay, the first step of mitosis is prophase. Prophase is characterized by the presence of chromosomes compacting, becoming more condensed. They're shortening. We can see this physical condensing of our chromosomes. Okay, the second step is metaphase. Okay, metaphase will begin to see that chromosomes are aligning towards the center of the cell. Okay, anaphase is characterized by these now sister chromatids starting to separate and move towards opposite ends of the poles in the cell. Okay, telophase, chromosomes are arriving at opposite ends. So we're going to have 2 distinct sets of chromosomes now at opposite ends of the cell. And cytokinesis, you're going to start to see the sort of indentation around these 2 sets of nuclei. And cytokinesis is going to result in the division of the cytoplasm to separate these 2 nuclei in order to get 2 daughter cells. Let's look at a representation of what's happening here. So we're still talking about the stages of mitosis, okay? Mitosis starts with prophase, where we have visible condensing of our chromosomes, where they start to shorten and compact, and we can actually see our chromosomes present. Metaphase, we're going to see the aligning of our chromosomes towards the center or middle. I remember metaphase with M. Metaphase, middle. These chromosomes are going to start to align towards the middle of the cell. Okay. Anaphase, we're going to get the separation of these sister chromatids. Okay, moving towards opposite ends of the cell. Okay, because the goal here is to create 2 cells from this 1 cell. All right, telophase, we're going to start to see these nuclei, 2 independent nuclei forming around these separated sets of sister chromatids. And then we'll start to see the indentation of the membranes here, preparing for cytokinesis, where this elongated cell now splits off into 2 new cells. And this is just another detailed representation of what's happening during prophase. All right. This process is taking place in somatic cells. All right, the process of mitosis, okay, often referred to as cell division or somatic cell division, takes place in order to generate new daughter cells that are going to be genetically identical to the parent cell. Okay. At the end of mitosis, we have two cells with the same structures and numbers of chromosomes as the parent cell. Okay, this is what you should understand. At the end of, once mitosis is complete, there are two new cells that have been formed. They have the same structure and same content. Okay, so we'll stop here.